Hello and welcome to Nikolai's genetics lessons, and this video is going to be about forensic science and genetics. And today I have prepared two multiple choice questions for you. This is very easy questions. I hope most of you would be able to choose correct answers. As usual, I recommend you to pause video here, try to find the correct answer, choose the correct answer, and when you would be ready you can run video again and you can compare your answers with my answers and explanations. So here's the first question. How much blood is necessary to make a DNA match? And three answers: a nanogram, a gram, an ounce. And you probably saw in many movies, especially old ones from 90s, where blood is taken from the vein. So pretty lot of blood were needed just a couple decades ago for DNA testing, but nowadays we can cross out this answer. So basically we have to choose between a gram and a nanogram. A nanogram is 1 raised minus 12 kilograms, or 1 raised minus 9 gram. So 1 billionth of gram. And this is about the weight of one cell. So answer A. Actually, none of these answers are correct. We just can say that answer A would be a better choice than answer B, because actually for DNA fingerprinting we need about 100 cells, 150 cells. With most recent and advanced techniques we may need between 20 and 50 cells. Still, this is not a single cell. This is just in theory. We can use one single cell, amplify it, and then we can do a DNA profile. But still, between these two answers, answer A would be more appropriate. But you have to understand that answer A is just approximation, but very close approximation. And next question, what is DNA amplification? And answer A, using sound waves to break apart the DNA into individual components. You may think that this is incorrect answer. This is true, this is incorrect answer, but actually sound waves or ultrasound were used in order to break DNA to small random pieces during, for example, human DNA project. Because scientists have to do thousands of tests of the DNA, for example, of the same fragment of, for example, the same chromosome, and they needed random fragments of random length. They cannot do it with restriction enzymes, because restriction enzymes would cut DNA in the same places, basically in the same places. But in order to sequence human DNA, scientists needed such breaks in random places, so they used sound waves or ultrasound. But actually we do not use this technique in order to amplify DNA. So if you think that by breaking large piece to small pieces we amplify DNA, this is not true. So we can cross out this answer. And now we have to choose between answer B and C. Answer B is the process of creating multiple copies of DNA from a tiny sample using the polymerase chain reaction, or PCR. And answer C, creating a computer model of DNA and displaying it on the screen so that it can be seen easily by a jury. Actually, answer C, such models or reports are present to the jury in order to, for example, exclude or include some biological evidence, for example, from suspect, and to the biological evidence that were found at the crime scene. But this is not what we mean by DNA amplification. So we can cross this answer also. And the correct answer is answer B. So basically, polymerase chain reaction means that when we take some fragment, DNA fragment, with the help of polymerase chain reaction, we produce multiple copies of this fragment. Because in many techniques is not so sensitive to work with single copy, so we need many copies in order to be able, for example, to detect bend on the gel. Copies that are similar in length, of course, would make one bend on the gel, and we wouldn't be able to see one molecule, but we would be able to see millions and billions of similar length molecules. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. And see you in the next video. Goodbye.